So I will share with you how we have built a national infrastructure for precision medicine, despite that we have a regionalized healthcare system. And in Sweden, we are lucky to have a national infrastructure for life sciences called SciLife Lab, which has 10 technology platforms that serves the country in genomics, proteomics, metabolomics, etc. And 10 years ago, we started to discuss with them, shouldn't we form clinical genomics units in order to transfer the new technologies? And this was quite successful, and we started clinical genomics nodes at all medical faculties in Sweden, which has the task of working between the hospital and healthcare, for instance, to set up clinical whole genome sequencing and gene panel diagnostics. And in this process, we thought, why not start a national initiative, Genomic Medicine Sweden, for upscaling and broad national implementation. And this research implementation framework proved to be quite successful in taking new discoveries or new technologies from SciLife Lab, adapting them for diagnostics through the clinical genomics nodes, and then validation and implementation upscaling through Genomic Medicine Sweden. So we have at each of the university hospitals, we have a genomic medicine center, and they are twin nodes between clinical genomics to use the the technology platform from SciLife Lab, and then the genomic medicine centers are at the university hospital. So this is a bottom-up initiative, and we have 7 genomic medicine centers, and so it's a regionalized infrastructure, whereas of course we harmonize on a central level regarding what type of reagents, what type of gene panels, how to interpret data, the pipelines, etc. And also the actual precision diagnostics and treatment is carried out at the university hospitals. We have one common IT infrastructure called the National Genomics Platform. And in this way, we can serve the whole country, despite that we have a regionalized healthcare system. We have 21 healthcare regions in Sweden, and all these genomic medicine centers cover a number of surrounding healthcare regions. And the National Genomics Platform is a natural resource. It's for both, of course, for healthcare, but also research. And this is located in the west part of the country. And currently, we have gone through the procedures, and we have now agreement, so we can share data across the country for genomics between the 7 genomic medicine centers within research projects where we have ethical approval. So our current legislation does not allow secondary use of health data. So there is an ongoing inquiry in order to enable this, similar to have been legal changes both in Denmark and in Finland. So this is a barrier right now, and hopefully the European health data space also will help us to move forward in order to enable secondary health. I mean, we are allowed to do secondary use for research, but not for the purpose of healthcare. So these are our focus areas. So it's rare diseases, 3 different cancer tracks, microbiology, pharmacogenomics, and complex diseases. And I will give you some examples of the activities. So we set up whole genome sequencing for rare diseases already 2015 at Karolinska University Hospital. And now there are 4 genomic medicine centers in Sweden that perform whole genome sequencing and serves the country. And this has gone quite rapidly. We did 5,800 patients in routine practice last year, and altogether 25,000 clinical whole genomes have been performed since 2018 when we started. And the success rate of the diagnostic yield is between 30 to 50%, depending on which type of rare disease that we are. You can see to the right different type of subgroups of rare diseases, and the percent figure is the success rate, the diagnostic yield. We are introducing whole transcriptome sequencing as an additional method, And we also are setting up a digital consent project. We have a newly developed app in order to get informed consent in a more streamlined way over the country. So this will hopefully make it more effective. For hematological malignancies, we have started with national hematology panels, and the first panel, it's a capture-based, almost 200 gene myelogene panel that we launched 2020, and is currently, last year we did something more than 4,000 gene panels across Sweden. So it has quite rapidly become the standard of care of the country. And I have to say that this has gone also quite smooth. The hematologists can receive this amount of data, and they can handle it, and they have become, I would say, experts in many of the different gene aberrations that can occur in hematologic malignancies. We are soon launching a lymphoid gene panel, and I will come back to this with whole genome and whole transcriptome sequencing. So we have performed a quite thorough validation in different phases. Of course, the first, the design, and established the SOPs both for the laboratory and bioinformatics. We have done an interlaboratory comparison and also validated at each lab before implementing it. So whole genome sequencing is what we think the next step for hematological malignancies. We did a retrospective study on 250 cases that show that we more or less can see everything we need to see in standard of care. And if you don't know acute leukemias, we do a lot of genetic tests. Today it's chromosome analysis. For pediatric leukemias, it's 8 different FISH analysis, it's array analysis, and a number of other target analysis. And we could see that we could detect all of them. And then we enter the prospective phase, where we aim to include 352 patients, which we have just recently passed. And currently we are going there. What we do is whole genome plus whole transcriptome sequencing. And so far, everything looks very good. We can see everything we need to see according to the guidelines, and also we get additional information in quite a substantial number of patients, and especially whole transcriptome sequencing it simplified the analysis for us, that we can rapidly also see fusion genes, which are common in hematology. For solid tumors, which is of course a much larger group, we have a 560 gene panel that the validation was finished last year, and now we have an ongoing implementation at all 7 genomic medicine centers. And the government has launched national projects for breast, ovarian, and lung cancer that we have conducted. Of course, we are also working on the liquid biopsy side. And this gene panel includes not only SNVs and indels, but also more complex markers like HRD, microsatellite instability, etc. And we also are part of an innovation milieu called Testbed Sweden, where we have the ambition to start new clinical trials. And there are many stakeholders within this milieu, not least agencies. And in this way, we hope to solve many of the hurdles for national precision medicine studies. So we just released the synopsis of a first national precision cancer medicine trial based on the drug repurposing protocol that you can see to the right. And hopefully this will start early next year, and we will have many different types of entries into the study. We of course want to use our GMS 560 panel, but it can also be other validated tests to be included in the study. We will also have the possibility to do pre-screening, that is to do the screening of potential biomarkers earlier in the disease, and then in order to plan ahead if the patient relapses again, and then can be included in this study. We will do an ambitious ctDNA analysis once a month, the first 6 months, in order also to follow the disease evolution. And of course, Drup studies are not only performed, I mean, there are several countries, not least in the Netherlands, but also other Nordic countries are performing already Drup clinical trials. And we have got 2 EU grants in order to both focus on precision diagnostics and precision treatment using the Drup protocol across Europe. So there are 15 countries involved. Dr. Stensinger is representing Germany in these 2 EU trials. And in my view, Sweden now is speeding up in order to be part also of data sharing on an EU level, because we, when we have our trial up and running, we foresee that we can, by sharing information between countries, and even hopefully in the future also sharing different baskets and treatment baskets, this will speed up the process of performing this type of studies. In childhood cancer, we have, since a number of years, been able to offer all patients with children with cancer whole genome sequencing and RNA sequencing and DNA methylation analysis. For brain tumors, we do the methylation arrays. And this has involved a lot of clinicians and laboratory people across the country. And we recently reported the first 118 patients where we saw that more than 90% had clinically relevant genetic alterations. We have finished the review of the first 300 cases now, and we see that 22 out of 300 were given targeted therapy. We do this analysis at diagnosis compared to many other studies that do it later in the metastatic situation, but we do it at diagnosis. Also, the review of the first 300, we see around 11% that has a predisposition to cancer. So currently we have come up to 500 cases, and we will continue, and we hope to turn this into routine practice for pediatric cancer. So this is the summary from last year, what we did. Sorry, I see that there are some Swedish, so it's rare diseases to the left, where we still do around 2,000 exome sequencing. No, that's the yellow one, exome sequencing, and whole genome sequencing, almost 6,000. We have a population of 10.5 million in Sweden. And then for solid tumors, we do almost 9,000 gene panels in the blue one, but we are now also using RNA sequencing, whole transcriptome sequencing, and whole genome sequencing. And hematology is still mainly broad gene panels. And these are the numbers so far that we, since we started 2018, we capture, or we follow these data carefully over the country in order also to see how the field is developing and the increase. And we foresee that there will be quite a rapid increase, not least for gene panels in cancer. Of course, we need to collaborate, and it has been fantastic to work with Dr. Stensinger and others within the ZPM network. We were quite early, we established a memorandum of understanding, and we have already had a few papers, and also we had an international symposium that we arranged in Stockholm last year. That was really a fantastic event. But we are also working together with Bettina and the National Genome Center, with France, Great Britain, and of course we are part also of the 1 Million Genome Project. And based on this symposium that we arranged last year in Stockholm, we have just published a European perspective on precision medicine, where you see that there are 7 countries that are described, where we sort of have tried to identify if there were top-down or bottom-up initiatives, and to showcase that you can build, even if you do it bottom-up, it's possible to create national networks. There are also 4 other studies. 3 of them are also accepted. So you can visit the Journal of Internal Medicine website to find these topic-specific reviews also. And of course, we need national and international collaboration, but also local, regional collaboration. And in Stockholm, we have established the Precision Medicine Center Karolinska, and this is the director of the hospital and the vice chancellor really decided that they wanted to make sure that precision medicine come into to the patients directly. And they sort of want to solve hurdles along the way. And this has been a very exciting way of working to change reimbursement models, how the workflow for patients, etc. So I think one shouldn't forget that even though it's important with national and international collaboration, you also have to do the work at the local level. So what we see based on our experience in Sweden, we think that even though we have worked a lot bottom up and enable a national network, we still need a top down, and we hope that there will be a partnership between the state and the healthcare regions, which are autonomous from the state in Sweden. And we want a permanent infrastructure for precision medicine. Of course, this with data sharing is key. If we want to, if we should be able to share between the countries, we have to be able to share also within the country. And we want to continue, of course, with the many different technologies. That's why we keep a close collaboration with SciLife Lab. And as I've already alluded to, we need to foster more national trials, and we have to also educate the profession, patients, and the society at large. So we will have a new symposium, the Nobel Symposium in September, also on precision medicine, that I invite you all to. It's fully booked, but there is also a possibility to attend virtually online. And with that, I thank you all for listening. And yes, I'm not sure if we are taking any questions or if that is done afterwards.